symposium. And also thanks for our organizers and dear parents for initiating, and also congratulations for this conference. So recent 20 years, the discovery of the many neurodegenerative diseases was done for recent 20 years, and I showed a couple of examples here. The Alzheimer's and Parkinson's and amyloid bladder sclerosis such as modern neuron disease. There are many genes, responsible genes discovered. And those diseases are characterized by the abnormal accumulation of the misfolded protein. And from the research, there are a couple of the major hypotheses on the neuronal damage was made, as I shown here. So, as I showed in the last slide, in the neurodegenerative disease, the mechanism of neuron death has been extensively studied. However, the many neighboring cells surrounding neurons are now shown to be involved in the disease process. So today I'm going to show characteristics and basic research of motor neuron disease ALS, and then through the ALS research by using rodent or laboratory animal, elucidating the active role of glial cells, the cells surrounding the motor neuron. And then I will show a couple of examples of the growing concept of this the role of glial cells in other diseases. Amyloid bladder sclerosis is characterized by the death of upper and lower motor neurons, which reside in the primary motor cortex and lateral side of the spinal cord. And it is typically a rate onset disease and rapidly progressing to death within 1 to 5 years after onset, which causes the general skeletal muscle paralysis, including the tongue and also swallowing difficulty, and eventually cause respiratory paralysis. And most of the cases are sporadic. 10% of the cases are familial cases. And currently, 8,000 patients are suffering in Japan, and I assume 10 times more patients are probably suffering in India, since the prevalence is almost same in the worldwide. So currently, no viable therapy is available. So there, I showed a couple of the famous ALS patients, find the Stephen Hawking and the very famous baseball players and the composers and also the politicians. So generally, the sensory system, eye movement, and the urination, and intelligence and the mind are not generally affected. So those patients are suffer from ALS, but mostly the intellectual performance are preserved. So what we learned from the neuropathology from human patients of ALS and the extensive growth, degeneration of the motor neuron with accumulation over time within the body is known as some protein accumulation. And here it may be difficult to see the difference, but there is a loss of motor neuron in the pink dot shown here. And also another feature is loss of lateral column axon, which is axons of upper motor neuron from a primary motor cortex. So we also learn from many things from the genetics of ALS. So initially, the responsible gene of the ALS is only one, SOD1, which was discovered in early 90s. But since then, there are more than 10 different responsible genes were identified. Yes. Yeah. Okay. SOD1, dominant mutations in SOD1 are most frequent cause of inherited ALS, and it accounts for 2% of all ALS cases. And SOD1 is an enzyme that detoxifies the superoxide radicals. It converts to the hydrogen peroxide, and with the help of other enzymes, eventually it converts to the water. And in the ALS cases, this protein is only 153 amino acid, very small proteins, and forms the homodimers. But with the introduction of the mutations, in the most cases, the single amino acid substitution, it causes motor neuron disease. So far, many researchers agree that SOD1 mutations provoke disease through a gain of toxic property, not loss of enzymatic activity because of those reasons. So some of the disease-causing mutations still have enzymatic activity. And also, the SOD1 knockout mice, so the mice deficient of SOD1, do not develop motor neuron disease. So in contrast, overexpression of the mutant in mice that produce the progressive motor neuron disease regardless of SOD1 activity. So this is a typical end stage of the ALS mice, showing the complete hindbrain paralysis and motor neuron degeneration. So in the cell level, mutant SOD1 form aggregates. Mutant SOD1 proteins provoke toxicity in the cultured neuronal cells. So this is the neuronal cells expressing mutant wild type or mutant SOD1 fused with green fluorescence proteins to visualize the SOD1 proteins. So these two apparently looks different, but the only the difference is the single amino acid substitution. It causes the completely change the form of the protein, which is so-called misshaped or misfolded proteins. It's really easy to form protein aggregates, and also it is also seen in the mouse model. So this is the typical end stage mutant SOD1 transgenic mice which carry the mutation derived from human patients. It shows difficulty in walking in hindbrain paralysis. Okay, so here very recently, and the SOD1 mutation is also found in dogs that is causative for degenerative neuropathy, but it is very similar to the human ALS disease. Okay, so this is the dogs affected in this disease and showing the hindbrain paralysis and showing the very similar pathology to human patients with SOD1 gene mutations. So in the advanced stage, the dogs has complete hindbrain paralysis. That's really a devastating disease like human motor neuron disease. Okay, so far, mutant SOD1 mediated toxicity has many hypotheses. And in the downstream, there are several prominent hypotheses. I just highlighted here protein degradation impairment, ER stress, mitochondria, or axonal transport. And today I'm going to focus on the glial cells. In terms of the protein misfolding, I just show you one slide of our collaborative ongoing project collaborated with Saitama University and also Dr. Biani to detect the mutant SOD1 like this. So we're going to try to identify the peptide aptamers which specifically recognize the mutant form of SOD1, which may be used for diagnostic or therapeutic application. So the idea was initially proposed from Dr. Biani, and now it is ongoing. And So apart from this toxicity problem, we have been focused on other important questions for many years, which is other cell types other than motor neurons involved in mutant toxicities. So since SOD1 expressed in all cell types, but causing the motor neuron degeneration. However, in the same time, the mutant also expressed in the cell types surrounding motor neurons. That's why we are very interested in the mutant action within those cells. So there are two different hypotheses to explain the neuronal degeneration. The one is the cell-autonomous neurodegeneration, and the other is non-cell-autonomous neurodegeneration. So in this case, so this particular motor neurons carrying mutant proteins are dying by the toxicity within its cells. So in this case, the surrounding cell does not influence to the fate of these motor neurons. So in the non-cell autonomous hypothesis, that this particular motor neuron is dying by the toxicity from the neighboring cells. So this is the concept of the non-cell autonomous neurodegeneration. To test cell autonomous hypothesis, we developed a new strategy to generate chimeric mice in which the mutant SOD1 is expressed in all motor neurons. So we took the embryo from the mutant SOD1 mice, which can contribute or differentiate to the all cell types. And we took the embryo from olive knockout mice, which cannot generate motor neurons. So olive is a transcription factors which are essential to differentiate to motor neurons. The mice without olive cannot generate motor neurons and cannot move and cannot survive. However, when we successfully make this chimeric mice, the motor neurons are all derived from mutant SOD1 embryo because this cannot generate motor neurons. And the different amount of other cell types are a mixture of wild type and mutant SOD1 expressing. So in other words, I'm trying to replace the surrounding environment with wild type cells of motor neurons. So we initially generate such kind of chimeric mice and confirm the expression of the mutant SOD1 in the motor axons and sensory axons. So in these axial sections, we check the expression of the mutant by the human SOD1 staining. And here you do see several green spots, which means the expression of the mutant SOD1 is very few. That means a chimeric of mixed wild type and mutant. So in contrast, the motor roots, almost all detectable axons express the human SOD1, mutant SOD1. That means our design of the genetics is actually working. So we follow up those mice and to see whether the motor axon degeneration is actually happens. So this control mice, which express mutant SOD1 in all cell types, that cause the significant or extensive axonal degeneration at 6 months. However, this only chimeric mice, despite of the mutant SOD1 expression in all motor neurons, the substantial number of the motor axons actually survive 2 months after all germline mice has reached to the end stage. So this really indicates the motor neuron degeneration itself is not cell-autonomous process, because the many of them still survive. Then we next asked whether the survival of such a mice has extended. So this is the one of the representative chimeric mice which really survives longer, about 2 months longer than the germinal mice. And we actually terminate those mice for histological analysis, but they still escape from motor neuron disease. They are really walking well and also climb up to the cage. So those results really indicates toxicity of mutant SOD1 is non-serotonomous. That suggests toxicity from non-neuronal cell is required to drive the motor neuron degeneration. So then next obvious question is to identify the non-neuronal cells which are targeted for the toxicity of mutant SOD1. So to identify such cell types, we actually generate the new class of the mutant SOD1 mice, which express mutant SOD1 in all cell types, but by using the Cre-Dox recombination technology, we are able to remove this transgene in the cell type specific manner when we cross with pre-mice. Then we may be able to test whether elimination of mutant in motor neurons or other individual cell types that really really slows the disease progression. So we initially generate those mice and also obtain those pre-mice from the collaborators. For example, the motor neuron specific pre-mice or astrocytes or mast cells and the microglia and the macrophage and Schwann cells. So initially we tested the motor neuron pre-mice to test whether the removing SOD1 from motor neuron is sufficient to halt disease progression. So removing mutant from motor neurons by using VChat pre-mice actually slows disease onset. This is the onset curve by 50 days. However, the survival time, the effect of the survival time is almost same. So that means there is no further slowing effect of disease progressions. This really indicates the disease onset is mainly determined by the toxicity within motor neurons, since we are able to slow the disease onset. However, the toxicity within motor neurons is not sufficient to drive disease progression. This was quite surprising, and the motor neuron is not only target for this disease. And those observations was also tested by the several groups, including ours, by using siRNA approach or other Cre-Dox recombination technologies. But all those studies reached the same conclusion. The mutant SOD1 in motor neurons actually determine the time of the disease onset. How about the non-neuronal cells? So we first focus on these two types of the glial cells, macroglia and astrocytes. The microglia is a central nervous system immune cells derived from the peripheral monocytes or similar to the macrophage. And this really remove the pathogen and also the clearing the dead cells. And astrocytes, so this is really supporting the action of the neurons, and also they have many factors, of course, supporting the survival of the neurons and also controlling the function of the neurons. So there are several evidence of the role of microglia and astrocytes in ALS. since the astrocytic or microglial activation and also the proliferation of those cells are seen in the region of the SOD1 mice and human disease patients. Elevated production of cytotoxic molecules and also inflammatory molecules was seen in the mouse model and human patients. However, there was no direct evidence that those glial cells are actively contributed to the motor neuron degeneration in ALS. So we used those mouse model and cross with microglia specific pre-mice to remove the SOD1 from microglia and macrophage, and surprisingly, the survival was nearly extended, almost 3 months. And this effect is mostly derived from slowing rate phase of disease progression. And in the same year, the group from United States actually reached the same conclusion to our results. So they have transplanted wild-type microglia and slowed the disease progression of other SOD1 mouse model. So these two studies really indicates microglia is a determinant of disease progression of ALS. We also tested the role of the astrocytes by using astrocyte-specific pre-mice and confirmed the specificity of gene expression. It may be very difficult to see, I'm sorry, and confirmed the specificity of the gene excision by a real-time PCR, and there is no excision in the primary microglia, but a substantial gene reduction in primary astrocytes when CRE is present. And this is the overall results, and motor neurons specific reduction slowing disease onset, and microglia and astrocyte specific gene removal slows disease progression. And detailed result also indicates there's no effect of disease onset when we remove the SOD1 from astrocytes. However, there is a twofold extension of disease progression when we targeted astrocytes. So those all studies really indicate the active role of glial cell in disease progression of motor neuron disease. So when we observed the symptomatic double transgenic mice which removed the mutant from astrocytes, the environment of more pre-positive astrocytes that marked with beta-tau staining, there is a less microglial activation. So we observed nice negative correlation between the activated microglia and pre-positive astrocytes. So that really indicates the mutant expressing astrocytes augmented microglial activation, then contribute to accelerate disease progression in ALS model. So the group from Johns Hopkins University, they transplant wild-type astrocyte precursor cell in ALS rat transgenic model and leads to the same conclusion. They slow disease progression. So I have presented 3 independent results to test the individual cell types, and we also test muscle and also Schwann cells. So for the muscle, we have 2 different approaches. The one is try to increase the muscle mass by follistatin, and second approach is again to remove mutant SOD1 from muscle by Credox technology. Since muscle is really easy target for in terms of the accessibility, then we try to use the myostatin, which is a negative regulator of muscle growth. So it is known that the myostatin knockout mice has increased muscle mass like this. Really, it has more muscle volume. And also, in the American group, they have very non-clever approach to produce the double muscle cow to obtain more beef. Okay, then we did the same approach, try to increase the muscle mass in the sodium mouse model, and we observed the more muscle here. However, there is no effect of disease survival of the mice of SOD1. And we have done another experiment, try to remove mutant from muscle. However, there is no effect on the survival of the SOD1 mice. So the muscle is not a target for sodium-mediated toxicity, at least in our models. Finally, we have tested the role of the Schwann cells, which is another important neighboring cells of motor neurons. So Schwann is myelinating glial cells at axons of peripheral nerve, and about 1,000 of the Schwann cells lack axons of only single motor neurons. So we have done the same experiment, try to remove mutant from Schwann cells. However, very surprisingly, it accelerates the disease course. And we are very confused by this result. But our interpretation is probably the mutant SOD1 or SOD1 enzymatic activity may be beneficial to the Schwann cells. So in summary, toxicity within motor neurons determines the time of disease onset, and toxicity within microglia and astrocytes determine the time of disease progression. And astrocytes enhance the microglial activation, and Schwann cell and muscle has no protective effect in terms of the SOD1 toxicity. And microglia and astrocytes play active role in disease, and they are a viable target for the therapy to slow disease progression. And more importantly, onset and disease progression of ALS are determined by actually different cell types, such as neurons and glial cells, to generate non-self-autonomous neurodegeneration to the in ALS model. And this non-self-autonomous neuron death concept is recently tested in other neurodegenerative diseases such as Alzheimer's and Parkinson's and polyglutamic disease. And all of those supports the good neighboring cells are very important. And finally, I'm gonna discuss about the stem cell research in ALS. So probably you know the name of the embryonic stem cells and the induced pluripotential stem cells like iPS cells. So in the ALS field, the very recently first iPS cells and motor neurons derived from iPS cells was established from the skin cells of 82-year-old ALS patients. So they really confirm the production of such a motor neurons from skin cells. And those similar kind of approach easily can be done by using a mouse ear cell, and it nicely differentiates to the motor neurons. However, I'm very ambiguous for application of this motor neuron, stem cell-derived motor neuron for clinical or therapeutic application, since the lumbar motor neurons in human requires to extend about 1 meter long axons, since our egg is much longer than mouse. However, they demonstrate the importance of slowing disease progression. So this graph simplifies the ALS disease onset and end stage, and this actually represents the disease progression from the onset time to the end stage. So, as I told you, the ALS and Alzheimer's and Parkinson's, the 90% of the patients do not carry the mutant genes. So that means the sporadic. So everybody may be affected by those diseases. So in this case, the patients only visit to the clinic after disease onset because we do not know who developed those diseases. Then, if successful therapy or viable therapy is available, then the realistic goal is to slow disease progression. So that's why to uncover the mechanism of disease progression in neurodegeneration disease like ALS is very important to identify such a disease-modifying therapy for those devastating diseases. So this is a summary and perspective that I don't want to refrain. And I'd like to thank to all collaborators and also the laboratory members to this presentation. And thank you for attention, and I stop here.
Thank you. So anyone?
Someone before started coming from developing nation, that the HIV patients, they're getting drugs, and they're getting better, they're living their life. And now they started having neurocognitive disorders. And no matter what you say, in case of HIV patient, there's a lot of oxidative stress, one because of the virus, further that's because of the drug they take. Do you think this kind of a mechanism is possible for the development of neurocognitive disorders?
Here, the next question. So, is the oxidative stress is?
Well, in case of HIV patient, from 2001 onwards, it has been noticed that the neurocognitive disorders has been increased many-fold nowadays among the common HIV patient compared to the common population. Of course, because the HIV patients, they can live longer. So, we cannot cure HIV. We can control HIV. So, that the patient continues with the HIV patient, apart from they take drug. So, chances are they induce a lot of pro-inflammatory cytokines and inflammatory cytokine. That may be responsible for the induction of oxidative stress. So do you think that could be a possibility for the neuroaids, and one of these possible mechanisms is there? That's a possibility?
Yeah, it is possible.
Thank you.
However, there are many mechanisms and also the combination of many possible mechanisms.
Thank you. Anyone from the students? Yes, please, I can see one hand. Can you link your question to the motor cell diseases that Dr. Yamanaka has been working?
Actually, the research was somewhat like that. The motor neuron, that they are supplying the ketone bodies to the motor neuron. Sir, the ketone bodies are being supplied to the motor neurons for preventing degeneration. And another protein, alpha-synucleoprotein, it has been also found in the brain for preventing the whenever the neurons, motor neurons are being degenerated, that particular protein, alpha-synucleoprotein, it has been found there generating itself. So, can you please explain a little bit about this?
Would you like to say something?
Yeah, it is really difficult.
Ask the question, but
Actually, the science direct.
please tell, you know the answer. Please
No,
tell us the
ma'am,
answer.
actually the science magazine was there, in that a research had come. So they had just given an abstract of that. They have found alpha-synucleoprotein in the brain. it is being found there, and that I asked the question. So I have not got actual mechanism of that, how motor neurons are being prevented from degeneration.
Would you like to say something?
I think the Newton made the toxicity how they amplitude motor neuron. There are combination of the toxicity. There are not only single pathway.